Hi, my name is Tyson Dawson. I'm a PhD candidate at the George Washington University, and this is the contents of my dissertation defense, and I've entitled this seminar, Characterizing Microbes and How They Impact Human Health. And I'd like to start off this presentation with a question, and that question is, what do you think of when you hear the word microbiome? And my goal throughout this presentation is to hopefully amplify and expand what you think of when you hear this word. And I think that a nice scientific historical anchoring point for us today is the completion of the Human Genome Project, or at least the announcement of the completion of the Human Genome Project. And this occurred in the year 2000, and President Bill Clinton, along with some of his scientific advisors and other world leaders, gathered in the White House to announce that for the first time, a draft of the human genome was ready for analysis. This is after multiple years and multiple billions of dollars spent on this project. And the scientific community, and indeed society as a whole, had a significant amount of optimism in terms of what this would mean for the world. Indeed, in his address, President Clinton said that genome science will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases, and promised that in coming years, doctors would increasingly be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer. So now, 23 years later, we can look back at this moment of scientific history and ask, what was the meaning of the Human Genome Project, and perhaps why didn't we see the complete fulfillment of all of the promises that President Clinton outlined here? And today I'd like to talk about one reason why we might not have cured all diseases and we might not be able to prevent or treat all diseases, and that has to do with this additional layer of complexity that perhaps wasn't as well appreciated or understood at the time of the completion of the human genome. And that's the fact that you are not one genome, but in a sense, many genomes, and that you have this secondary reservoir of genomic diversity in your microbiome. Your microbiome is this collection of microbes, including bacteria, fungi, archaea, viruses that live on and in you across multiple niches. And it turns out that these microbes play an important role in the development of disease, in terms of how your body metabolizes different agents, in terms of your response to drugs, in terms of immunity. And indeed, if you were to count all of the cells associated with your body, 90% of them would belong to your microbiome. And this is possible because these cells are so much smaller than our own cells, but it speaks to the fact that this is an additional layer of complexity. And so I'd like to point out one particular area in which the microbiome plays a role in our health, and that is in the chemical transformation of these things that are called bile acids. It turns out that there are these metabolites, these molecules, which are produced in the liver and which make their way down into the intestine. And there, they are chemically altered by microbes that live there to become these secondary bile acids. And it turns out that these secondary bile acids are especially important in the development of metabolic disease and in things like obesity and type 2 diabetes because they affect the way our bodies digest and absorb fat. So this is one specific example of how our microbiome contributes to our health. It's through these biochemical pathways that are not encoded by our human genome, but rather are encoded by our microbiome. And so as part of my dissertation, we wanted to look at the role of these bacteria in the gut. We wanted to see how the bacteria in the gut play a role in digestive processes, and also how the bacteria in the gut, or specifically which bacteria in the gut, are involved in the production of these secondary bile acids. So with those goals in mind, I want to give you the outline of how this study was conducted. We performed a secondary analysis on an already published study where they had the following study design, and they did their own analyses, and we took this same data and did some additional analyses and published on it. The basic study design was that there were 22 patients divided into two arms, and those patients had their stool sequenced metagenomically at the beginning, before the start of the trial, and then after 4 weeks. Half of the patients, so 11 of the patients, over the course of 4 weeks, were administered what is called a fecal microbiota transplantation in the form of a pill. And this pill essentially contains fecal material from a single healthy lean donor. And all of the patients in this study are patients who are obese but do not have metabolic disease. And so the idea is that maybe you can take this healthy person's gut microbiome and transplant it into an obese person, and that the microbes that are present in the healthy person's gut, in the lean person's gut, can change the landscape of the person who's taking these pills orally. And then half of the patients in the trial, instead of actually receiving this fecal microbiota transplantation, or FMT, they instead just took a sugar pill over the course of 4 weeks. And so what we wanted to do, again, was to identify which species are present before the administration of FMT and then characterize it after. And we can also do some other statistical comparisons. So not only can we do before and after FMT, but we can do between week 4 of the patients who received FMT and week 4 of the placebo, in which another comparison, which we would expect to see some changes. And we can also do comparisons within the placebo group between week 4 and the baseline. And if there are changes there, then it probably means something went wrong with our study, because this is kind of a negative control. We don't expect that taking a placebo pill over the course of 4 weeks would have any significant effect on the gut microbiome. And so, again, these are the kind of 3 different kinds of analyses we can do. Again, before and after treatment, between treatment and non-treatment, and between placebo before and after. And we can do statistical tests to identify which bacteria, which pathways, and we also were able to characterize the metabolites that were present in this stool, specifically those secondary bile acids, so that we can identify which bacteria are correlated with the production of which secondary bile acids. So with this in mind, let me give you a brief overview as to what exactly we do with this data once it comes off of the sequencing machine. I'm a purely dry lab or computational biologist, which means that my work is exclusively on the keyboard and on the computer. So I wasn't involved in the generation of this data or in the collecting of the samples, but I was involved in developing tools and pipelines that help us analyze this kind of data in order to answer biological questions. So one of the first tasks was to characterize microbial diversity. And by that, I mean you take these reads, these metagenomic reads, which come from the bacteria in the gut, and you can identify which taxa are represented, which species, which genera, which orders and families are present in this data. And we had a tool in the lab that's called Pathoscope, and that tool has a great functionality. It uses this Bayesian reassignment framework to take reads and assign them to taxa, but it lacked some downstream capabilities. And so we extended this tool and also renamed it in the process to SeqSite. And so not only did we take these reads and use SeqSite to assign them to these taxonomic bins, but we also took reads and assigned them to gene families. These are clusters of genes with sequence similarity. And we could then use annotations about these gene clusters to conduct statistical analyses, specifically a pathway analysis. And we used a tool called OwnPath to do that. And then we're also able to take these metabolites, specifically these secondary bile acids and the concentration of these bile acids, and do a correlation analysis or association analysis to identify which bacterial species are involved in the production or associated with the levels of which bile acids. And so on the right-hand side of these, I recognize that these are probably kind of small, but I wanted you to kind of get an idea of kind of the workflow of what we're doing with the data to transform it and get answers to our biological questions. And then in this next slide, let's actually look at some of these results that are hopefully big enough for you to see. One of the questions we had initially was which digestive processes are changed by the administration of this fecal microbiota transplantation. And so here I'm showing 2 of these comparisons. On the left-hand side, you'll see FMT at week 4 versus FMT at screening. So these are patients who received the treatment. We're also doing the comparison between people who received the treatment at week 4 versus people who just received the placebo at week 4. And then that third statistical analysis that I mentioned between the placebo group between week 4 and baseline, as we expected, turned out to be non-significant, so I'm not presenting that here. And don't worry too much about the dots and their orientation. I want you to focus on the names that are here on the left-hand side. These are pathways which are differentially enriched in these patients. So these are biological changes that are occurring in the gut that we can observe from mapping these reads to these gene families and doing statistical analyses. So one of the first pathways is this polysaccharide transport pathway, and that has to do with the intake of fibrous material into these bacteria. And it turns out that the bacteria in our gut can help us digest things that we can't digest on our own. And that includes taking fibrous materials, fermenting them, and doing digestive processes in that way. So that was significantly affected by the administration of FMT. speaking to how changes in the gut microbiome landscape can affect the way we digest our food. Another process or another pathway which was significantly different between FMT and before and after the FMT treatment, or between the FMT treatment and the placebo group, was this lipopolysaccharide biosynthetic process, which I'm highlighting here. And lipopolysaccharides, it turns out, are these bacterial toxins that can engage with the host immune system. And so we're observing changes in the levels of those as well, or in the processes that underlie the creation of these lipopolysaccharides. And then finally, we wanted to go back to this question, which is, we know that these bacteria create or are involved in the biochemical pathways that change these bile acids from the liver into secondary bile acids. And we wanted to see which bacteria in particular are associated with these changes. And we identified 5 that are kind of on the right-hand side of this heat map. And you can read those names. And then we're correlating these with these 10 different bile acids, which are shown at the bottom. And so the cells indicate, the color of the cell indicates the direction. If it's a reddish color, then that's a positive association. If it's a bluish color, then that's a negative association. So for example, in this first row, you see this Phocaeocola dori, and it turns out that that has a significant, illustrated by these stars that are listed in there, and it's quite significant. There's multiple stars here. There's a significant association between the levels of this bacterium and this secondary bile acid CDCA, and that's in the positive direction. So the more of this bacteria we observe, the more CDCA we observe. And so now researchers can take these 5, we've narrowed down this list from hundreds of bacteria that are present in the gut to this list of these top 5, which are associated with the production of secondary bile acids. And so what do we learn from this section? Let's revisit this question from the beginning. What is the microbiome? Well, it's this secondary source of genetic information that contributes to your overall health. It turns out that the sum total of the gut bacteria in your body encode hundreds of times more genes to perform these biochemical transformations than your own body does. And so we rely on these bacteria to help us with everyday processes like digestion. And so you are more than your genome. When I originally asked what your conception of the microbiome was, it's possible that your mind went immediately to the gut microbiome. It's arguably the most well-studied of all these microbial niches. And one niche which your mind probably didn't immediately go to is the nasopharynx and this nasopharyngeal microbiome. And it turns out that there's an area between your nose and your throat. You might be acutely aware of where this area is if you've ever gotten one of those really deep swabs for SARS-CoV-2, the agent that causes COVID-19. And it turns out that this niche is especially important in immunity and in immune response against respiratory viruses like COVID-19 or like SARS-CoV-2, the agent that causes the disease COVID-19. And it turns out that the microorganisms that live in this particular niche are friends and allies in fighting off these viral invaders. And these bacteria are invested in maintaining equilibrium and a homeostasis in this mucosal layer. So we had this hypothesis that perhaps in those patients who develop severe COVID-19, there is a dysbiosis or a change that's occurring in their nasopharyngeal microbiome, and perhaps we could characterize what that change is. So we were able to get our hands on some data. This is from the year 2020, March to June of 2020, when these samples were collected. We collected samples from 115 patients across the state of New Jersey. These patients were largely white and Hispanic and over the age of 50. Again, these patients had a nasal swab introduced into this nasopharyngeal area, into their nasopharynx, and RNA was collected from that area, and some of that RNA came from the virus, and so we were also able to characterize the virus as part of this analysis, and then we were also able to align some of this RNA to the bacteria and do what's called a metatranscriptomics analysis. So you're looking at what bacteria these RNA reads are coming from. And again, we're mapping using Pathoscope, or this new version of Pathoscope called SeqSite. Down at the bottom over here, you'll see that we are characterizing. We essentially compose a matrix of the microbial abundance of these different taxa across our samples. And then we do some downstream analyses, which I'll introduce you to here. One of the first things we wanted to do was we had a lot of metadata from these patients. What I mean by that is we knew a lot of clinical information about them. We knew their age and their sex. We also knew, for example, their BMI, different comorbidities they had, clinical parameters like levels of C-reactive protein or bilirubin. We knew which symptoms they developed, like having a cough or a fever. And we also knew some of these outcome variables, like how long were they hospitalized or how long they spent on a ventilator. I want to focus on that last one because to us, it's a good marker of disease severity, meaning that if you spend 0 days on a ventilator, you don't have particularly severe disease, but the longer you spend on a ventilator, the more severe COVID-19 you have developed. And so I'm showing this heat map here, which has these top 50 associations between bacterial species abundance and these clinical variables that we've measured. And I'm highlighting a particular row in a particular column. That column is ventilator duration measured in days. And a blue color in this column means that there's a negative association. So as you spend more time on a ventilator, you observe less of a bacteria. But there's one bacterium here for which we observe that the longer you spend on a ventilator, it seems the more of this bacteria you have. And that bacterium is this Haemophilus haemolyticus. And in the literature, if you look up this bacteria, it turns out that it is a bacteria that's associated with long hospital stays, invasive surgeries, etc. So this kind of presents a conundrum. Is this bacterium and its abundance upstream of the development of severe COVID-19 and it's causative in some sort of way? Or is it simply an effect of having to stay a long time in the hospital and having a weak body? And so in an attempt to answer that question, we wanted to go back to this idea of these immune system microbiota alliance that exists and that we know from the literature exists in this particular microbial niche. So much like we did in the FMT analysis, we took these genes from bacteria, these transcripts in this case, and we aligned them not only to figure out what taxa were present, but also to this resource called GMGC, the Global Microbial Gene Catalog. And this is a reference database that's well annotated for bacterial function, and that those bacterial functions include immune processes engaged in by the bacteria. So that's what I'm showing in this heat map here. Along the bottom, you have this list of these different immune processes, and along the right side, I'm listing some of these clinical variables. There's a lot of significant associations here, but let's focus again on ventilator duration, one of these markers of disease severity. And the color of the cells indicates sort of the direction of the pathway. Red meaning that that pathway is overexpressed, and blue meaning that it's underexpressed, or there's a negative association. So in patients who spent a longer amount of time on a ventilator, they had a correspondingly, there's this negative correlation with the gene processes related to immunity. So as you spend a long amount of time, the longer you spend on a ventilator, the more likely you are to have a downregulated bacterial immune function in this particular microbiome, in this particular niche. So it seems like what we were able to conclude from this is that the part of the development of severe COVID-19 seems to be related to a diminished capacity of the bacteria in the nasopharynx to participate in this immune system alliance and to help our bodies fight off against this respiratory virus, SARS-CoV-2. And so what's the big takeaway from this section? Let's go back. How should I think of the microbiome, or what does this word mean? Well, the microbiome can often be kind of a battlefield where you have multiple domains of life competing or working together in some cases. Here I'm showing an example of how the host immune system works together with bacteria to maintain the integrity and equilibrium of this mucosal membrane. And when these things go awry and when bacteria aren't expressing these genes and can't participate in this battle, it turns out that this can take a negative turn in terms of the health of the host. So there's these complicated interactions which we can measure with omics technology, and you should think about the microbiome as a place of different interaction networks that may include things like viruses, bacteria, as well as the host. So next, I want to pivot a little bit and talk about an area of the microbiome that you probably haven't considered before. And in order to do this, I want to introduce you to this fascinating and brilliant female scientist named Barbara McClintock. And she was interested in this pattern that you can observe in this corn that she's holding in her hand, this mosaic color pattern. And as she studied the corn genome, she identified that there were these genes that jumped around in the corn genome. And occasionally these jumping genes moved into or jumped into a gene encoding the pigment of a particular kernel, thus changing its color. And so she pioneered the research in this field of these transposable elements or jumping genes, and she was awarded the Nobel Prize for her research. And one of these classes of jumping genes are these things which are called ERVs. And I want to talk about these ERVs. And ERV is an acronym that stands for endogenous retrovirus. And when these things occur in humans, we like to call them human endogenous retroviruses, or HERVs. So I'll be using that acronym throughout the rest of the presentation, so keep its meaning in mind. A HERV is a human endogenous retrovirus. So if we take this word, we probably know what a retrovirus is. It's something like SARS-CoV-2, or it's something like a virus which causes the common cold, for example. They are these biological agents that are able to take their RNA, convert it into DNA, and stick it into a host. And it turns out that our ancestors, over the course of millions of years, were occasionally infected with retroviruses, but sometimes these retroviruses managed to lodge their genetic sequence of information not into a somatic cell, but into a germline cell, like sperm or egg. And thus their sequence became embedded into this evolutionary flow of information and was passed along to successive generations. And so it turns out that this happens so frequently, and replication events of these sequences happen so frequently, that today about 8% of the human genome is made up of these sequences which are of retroviral origin, these HERVs. So I kind of have an illustration on the right, thinking about how our ancestors, which were primate-like, we share this common ancestor with primates, how retroviral sequences inserted themselves into the genome and made their way to us today. And you might think that 8% is kind of a small number, but it turns out that only about 1 to 2% of our genome is dedicated to protein coding genes. And so the fact that we have 8%, this huge volume of genomic information dedicated to making viruses or storing these viral fossils, is quite interesting. And it poses a question of why have we kept these things around for so long that maybe we can talk about a little bit later. But for the first time now, and I'll talk about how we're able to do this, we can quantify the expression of these human endogenous retroviruses, and we can figure out how they are differentially expressed in disease. And part of the reason why we might be interested in doing this is it turns out that these are attractive candidates for studying autoimmune diseases. So the word autoimmune, or immune against the self, is characterized by this loss of tolerance, where the body loses its ability to figure out what is self and what is non-self. And what could be more paradoxically self and yet not self than one of these endogenous retroviruses? It lives inside of our genome, and yet it looks like a virus. It came from a virus. And so you can imagine that this could pose kind of a conundrum to the immune system and could be a target for immune activity that would then be targeted against the self, i.e. autoimmunity. And there are sort of two hypotheses for the role of these HERVs in disease, and I'll talk about them briefly. Both can be true, and some third thing and some fourth thing could also be true. Let's focus on these two for now. One of the hypotheses is that these HERVs contain functional enhancers and promoters that can change the expression of these genes. The idea here is that we've essentially kept these sequences around because they allow us to regulate our own gene expression. So we're taking or co-opting these sequences that came from viruses and using them to turn on and off our own genes. And that perhaps in disease, the activation of these HERVs become dysregulated, and we observe that in a disease phenotype. And the other hypothesis I kind of introduced in the previous slide, which is this idea that the HERVs so closely resemble or look like at a molecular level a virus that they act as an object or they act as an agent for the immune system to engage in an immune response against self. And this is this idea of molecular mimicry, that these things mimic at a molecular level what a virus might look like. So those are two potential hypotheses for the role of HERVs in the development of disease. And I mentioned earlier that we're able to quantify the expression of these HERVs. Turns out that HERVs are transcribed just like genes are. But for a long time, it was difficult to quantify their expression in a locus-specific manner, i.e. figure out exactly the genomic coordinates of where a particular HERV transcript came from. And I'll provide kind of an object lesson for why this might be difficult. Imagine you have a puzzle that you're doing, and there's a nice city skyline composed of many distinct buildings, and then there's a nice bright blue sky in the top part of it. And it might be easy to put together the buildings because they are of distinct styles, and there's sort of clear delineations between them. And so that part of the puzzle might be easy, but the top part with the clear blue sky might be difficult because perhaps it's all one shade of blue. Something similar is happening when we have to map these HERVs to the genome. The genes are like the city skyline. They are relatively different from one another, but HERVs are mostly similar to each other, mostly repetitive, and thus hard to map to the genome. So we use this tool that we've developed in the Computational Biology Institute called Telescope, which can take RNA sequencing reads and do a locus-specific expression mapping of these reads to coordinates in the genome, and it does so by employing a Bayesian reassignment framework. For this particular study, for which I'm about to present some data, we used, or about to present the result of some analyses on some data. We analyzed 2 data sets. These are data sets collected from the whole blood of patients who have systemic lupus erythematosus, a prototypical autoimmune disease which mostly affects women. It is associated with arthritic joint pain, with organ failure even. It's a debilitating disease. And so we identified 2 data sets where they had done RNA-seq on both healthy controls, so people without disease, and then people with lupus. And I sort of have a schematic down below of how this analysis, or what this analysis might look like. You have these databases that you're able to retrieve and get the sequencing reads from. In B, I'm showing how you would take these reads and align them to the reference genome. And here we're aligning both genes and HERVs, genes using a tool called STAR, HERVs using this tool called Telescope, which I introduced in the previous slide. You can compose matrices of expression of different transcripts across these samples, and then you can do some downstream statistical analyses on them. So let me show you the results of those analyses now. So we analyzed these 2 data sets separately, did differential expression analysis, and we identified 400 different HERVs, and that's if you take the union of the 2 data sets. If you take the intersection, it's a smaller subset, about 70 HERVs, which are differentially expressed in lupus patients. So some of these are upregulated, most of them are upregulated, and some of them are downregulated, so less expressed in lupus. And we wanted to tie this in with the gene expression results as well. We identified many genes which were also differentially expressed, and we had this question of, are there pairs of genes and HERVs whose expression is tightly correlated? Perhaps there are genes that turn on HERVs, or HERVs that turn on genes. And so we use this tool called Arboreto to find those gene-HERV pairs which had the tightest and most statistically significant correlations. And we took the top hits from that, and we wanted to visualize it using what's called a Circos plot, so you can see the chromosomal location of these elements. And we drew a line between these gene and HERV pairs. So you might be able to see that a lot of these lines, if you can see my mouse here, a lot of these lines seem to kind of go out and then stop. You know, we're drawing a line between these genes and HERVs. So what's going on with this line? Why does it stop? Well, if you really zoom in, you can see that it's actually kind of a hairpin, that it goes out and then it comes right back next to where it started. So it seems like physical proximity on the chromosome of genes and HERVs has something to do with the shared expression of these elements. But this isn't true of all of these gene-HERV pairs. I'm highlighting one of these associations with this red line that you can see. Some of these act at a distance, at a genomic distance, where these things, the HERVs and the genes, are located at physically distant parts of the genome. And so maybe we can dwell on that a little bit later, but certainly proximity of HERVs to genes has something to do with perhaps driving aberrant gene expression in lupus. And we also approached this in an orthogonal way and found something very similar. We wanted to see which pathways were differentially enriched in lupus. So we took the genes, which were differentially expressed, did an overrepresentation analysis, and we show those here. Don't worry too much about the color or orientation of the circles. I want you just to look at kind of the names of these pathways, and you see things like innate immune response, myeloid leukocyte activation, etc. And then we asked the question from a different lens, which is, let's take these HERVs, which are differentially expressed, take the genes, which are right next to them if there are such genes, and then let's put that list of genes into an enrichment analysis and see which pathways pop up. And it turns out that you get a very similar, if not essentially identical, set of biological processes, things like innate immune response, and you even see things like leukocyte activation, which we saw here before. So you're getting these same pathways, not only by looking at the genes, but looking orthogonally at the genes which are next to differentially expressed HERVs. So certainly, the physical proximity of HERVs to genes has something to do with the dysbiosis, or with the aberrant gene expression that we observe in this disease state. Which leads us to a question, kind of a chicken and egg question, of which came first, the gene expression or the HERV expression. Is it HERV expression that kickstarts the genes, which then maybe turn on the HERVs, and then you observe this positive feedback loop? This is something that's been talked about in the literature. perhaps the locus or certainly the locus-specific expression of these genes next to herbs wasn't well characterized until we did this analysis. But this is something that's kind of been known to be a problem, is how do you disentangle the herb and gene expression? And maybe there's even some third thing that kickstarts the whole process. And so in the remainder of the presentation, I want to dwell on this a little bit and show you some of the things that we've done to try to disentangle this complicated relationship. And in order to do that, I need to kind of introduce you to this idea that herbs and their nucleic acids are potentially immunostimulatory. There's been a lot of research in this domain that these herbs intracellularly can form this double-stranded RNA, these double-stranded RNA molecules. And these are typically characteristic of a viral infection. And so our body has a number of sensors that can pick up on this double-stranded RNA, including RIG-I and MDA5, which I'm showing here. And that upregulates or stimulates an interferon response, which is one of the hallmarks of lupus. So perhaps it's the capacity of these herbs to form double-stranded RNA that engages these sensors and upregulates interferon. There's also some other sensors or things that interact with double-stranded RNA within the cell. One of those is called ADAR. This is an RNA editing protein, which edits adenosines to inosines and changes the conformation of double-stranded RNA. It's inhibitory in nature, so an edited molecule is likely to no longer interact with MDA5 or RIG-I, and thus kind of you break this cycle if ADAR steps in to edit some of these. And then there's another type of nucleic acid, which is this zDNA or zRNA. It's a nucleic acid of kind of a different or flipped conformation. And there's a protein called zDNA binding protein, which can bind to this type of nucleic acid, recognize it as sort of as potentially pathogenic in nature, and turn on pathways like RIPK3, which can induce apoptosis and necrosis, so cell death. And so these are a number of these nucleic acid sensors which exist intracellularly, and our hypothesis is that herbs of different evolutionary origin perhaps possess different capacity to stimulate these sensors. And so we wanted to explore this, but first we had to overcome this hurdle of the fact that these herbs have names and sort of evolutionary classifications which aren't particularly actually rooted in true evolution or data science. I won't bore you with all of the details, but suffice it to say that some of these elements have been put into groups or given names based on relatively arbitrary trivia, to say it like that. For example, there's this family of herbs called Herb K, and it's named such because all of these things have a lysine tRNA, where lysine's single letter amino acid code is K. So anything that has a lysine tRNA is called Herb K. And sometimes these things also get their name because of a neighboring gene, an amino acid motif, a clone number, things which don't really speak to their true evolutionary history or origin. So I have this little comic on the right where the scientist is looking, I guess pretend he's looking at a herb or something, and he says, "I shall call you GERV, goggles endogenous retrovirus, since I found you wearing my favorite goggles." And sometimes to me it seems like the naming or putting into groups of these herbs is just as arbitrary as what this scientist is doing here. So here's what this database looks like. This is, again, this legacy database that we've been we've been handed by scientists that came before, where some of these names perhaps don't have a particular bearing on evolution. Some of these families have over 2,000 members. Others have much fewer, 6 members or 2 members. These are sorted by size. And on the y-axis, you have the frequency, and the bars are annotated by the size of a particular group. And there are these 63 herb families that have been recognized in the database that we use. And I wanted to rethink this classification basically de novo. And I wanted to say, let's throw this old classification to the side, and let's do a new classification system based only on evolution and data science, and throw everything else out, essentially. So what I did was I aligned all of these sequences using a tool called MAFT. I calculated phylogenetic relationships, and then I fed that into a clustering software called OhmClust, which can break these things into groups of shared characteristics, which again, our assumption is that these things then have shared evolutionary origin and or sequence similarity one to another. This resulted in 1,799 clusters, but a lot of these clusters were just sort of singletons or clusters that only had 1 or 2 members. So we imposed a threshold that we only wanted to consider those clusters which had a size of over 30 members. This yielded 75 clusters, which gives us a nice comparable number to compare to the 63 that I mentioned in the previous slide. So what is this? If we compare the old system versus the new system, that's what I'm doing in this heat map. The old names are on the right, and then our new classification system is down on the bottom axis. Our new classification system doesn't have particularly interesting names because the software doesn't know anything about these clusters. It's composing them de novo, and so it just gives them a number. So I want to highlight one example of, this heat map shows the overlap between a given cluster from our data science and evolution informed approach versus the legacy clusters. And the depth of the blue color indicates the proportion of HERVs that are shared in this particular group. And so, for example, this HML3 family was sort of independently recovered by our clustering approach and overlaps completely with our cluster 7. So this is actually a legacy family, which appears to be well supported by evolution and data science because we recovered it independently. Here's this other family, this HERV-H family, named because they all use this histidine tRNA, where the H is this one-letter code for histidine. And our approach grouped this into 7 different clusters. So based on evolution and data science, this large family is better broken up into 7 individual clusters. And then there are some families which are so hopelessly spread out across so many different clusters that it's hard to this sort of speaks to the fact that this family system is sort of outdated and not particularly based in evolution and data science. So I mentioned earlier that our reason for doing this and for creating this new classification system is we want to see if there are certain clusters informed by evolution and data science of HERVs that differentially interact with sensors of innate immunity. And that's what I'm showing here across these violin plots. And let me explain the plot a little bit to you. Along the y-axis, you have a prediction of the interaction of a given RNA molecule, and in this case, RIG-I, one of these stimulatory nucleic acid sensors that can turn on MAVs and induce interferon. And the score from 0 to 1 is basically, it's using this tool called RPI-seq, and you can think of it as a score of 0 means not at all likely to interact, and a score of 1 as 100% likely to interact. And then in each group, I'm using a red dot to show the in-group median of the interaction prediction. The blue dashed line indicates the median across all HERVs, and then we have a nice negative control all the way over to the very right, which is 10,000 randomly selected genes as a group for comparison. So genes, it seems, are not likely to interact with RIG-I. This probably means that they're not likely to form double-stranded RNA and have a conformation that would yield them to be recognized or to interact with RIG-I. But there are some groups of HERVs which do appear to have a greater affinity for interacting with RIG-I. One such group is here that I'm highlighting with my mouse, this third from the left. This median value is significantly higher than, for example, this group, which seems to be unlikely to interact with RIG-I. And so we've done this analysis not only for RIG-I, but for the other 3 nucleic acid sensors as well. One of those is MDA5, another one of these stimulatory sensors that can stimulate the production of interferon. And again, we see group specificity to some degree, that there are some groups which are particularly below, or their in-group median is significantly below that global median. Again, genes are not particularly likely to interact with MDA5. And then the same can be true of ADAR, which I'll show briefly, and zDNA binding protein. And we see group specificity. So there are some groups which are likely to interact with RIG-I, but not likely to interact with MDA5, ADAR, or ZBP. And that's true of all of these things. There's some which are likely to interact with ZBP, but not with the other 3. And so our hypothesis here is that this has to do with shared evolutionary characteristics within a particular group and sequence similarity, which would render them more or less likely to engage with these sensors of nucleic acids. And so we wanted to tie this in with cytokine stimulation. I mentioned before that there's this complicated relationship between cytokines like interferon and other cytokines and the upregulation of HERV elements, and potentially there exist these positive feedback loops here as well, where the interference stimulation upregulates HERVs, which have a capacity to engage some of these nucleic acid sensors. I'm showing you a heat map here, where along this axis, we have these different cytokine stimulations, and here we have these different sensors of innate immunity. And what we're analyzing here is we've identified those HERVs which are upregulated by particular cytokine stimulations, and we're finding a correlation between the log2 fold change of HERVs in that by that given stimulation and the interaction prediction with a given nucleic acid sensor. So I'm illustrating that in these two the additional panels in panels B and C, which are sort of illustrations of here. This is the interaction prediction with ADAR and tweak IL-17A stimulation. So this is this cell in the heat map visualized as a scatter plot with a regression line. And those HERVs, which are more upregulated by this particular cytokine stimulation, are also HERVs which seem to interact with ADAR. And so we're hoping that this kind of analysis and looking at the different evolutionary history of these HERVs and their different evolutionary origin and shared sequence characteristics, and determining what cytokines upregulate these different HERVs and what immunostimulatory capacity they have, that by characterizing these things, we can start to disentangle this complicated web of interactions that happens and the pathogenic nature of these HERVs within the cells. And so to zoom back out, I want to revisit this question from the beginning again, which is, what is the microbiome? We've talked about the microbiome in the gut. We've talked about the microbiome in the nasopharynx. And now we've talked about the microbiome even in our cell's most sacred space, this nucleus, this was supposed to be this fortress of solitude. We have this whole cellular compartment dedicated to containing the genome and keeping it safe. But it turns out that this is also the microbial domain. It turns out that viruses have colonized this space of our bodies as well. Indeed, 8% of our genome is composed of sequences of retroviral origin. These sequences are differentially expressed in disease, possibly involved in positive feedback loops. Their proximity to certain genes and pathways seems to be important in disease, but there are also these distal relationships that perhaps have bearing on immunostimulatory capacity, the ability to interact with these nucleic acid sensors, and that all has to do with the evolution and origin of these HERV sequences. And we're starting to kind of parse what's going on in terms of cytokine stimulation, upregulation of HERVs, and the immunostimulatory capacity of those HERVs. And there have been 3 publications that have already been accepted and published as part of this work, and I'm highlighting those here in case you want to take a look at those. And finally, I'd like to acknowledge my committee members who I have here in the center who have helped guide this work from the beginning, as well as collaborators. Science is a team effort. It takes a village to do these kinds of analyses. There are people who collect the data and generate the data. There are participants in these studies. There are people who have helped me with the analysis of the data, who have helped with biological interpretation, and I'm highlighting some of those collaborators here on the right. And I'd also like to thank the other members of the Computational Biology Institute for their camaraderie and friendship over the years and for their assistance in these projects. And with that, I'd like to say thank you.